Okay, so the prokaryote and eukaryotic cells contain the DNA as a genetic material. You know that in eukaryotes, DNA is organized as a distinct structural entities called chromosomes, which are present in the nucleus. Each chromosome is made up of DNA tightly coiled in many times around the protein called as histones. histones. So the number of chromosomes varies from two, sometimes many is also there. In prokaryotes, single which is closed and circular DNA is present. The naked DNA is present freely in the center of the cell, which is not cover any nucleus. So, what is the chromosomes here? The chromosomes are first described by the Strasburger, and laterally, which is observed the so X. So the term chromosomes was introduced by the Waldeyer and Sutton and Bavary is found the chromosomes act as a messenger of the heredity. The morphology of chromosomes was described by the Gaiter and uh, Kaufman. The chromosome number. The number of the chromosomes is the constant of the particular species. Therefore, these are the a great importance in the determination of the phylogeny and taxonomy of the species. The somatic chromosome number, the chromosomes found in the somatic cells, the plants and animals, which are represented by 2A, whereas the gametic chromosomes number is one of the somatic number, that is the haploid number, which is the N. So the haploid set of the chromosomes is known as a genome. The size of the chromosomes differ greatly in size in different organisms. Also present different pairs of the chromosomes in nucleus of some. Same cell have the different sizes. Metaphases chromosomes are useful to identify the size of the chromosomes. The plants have lower chromosome number and possess the longer chromosomes than those who have higher chromosome number. Dicots generally possess the high chromosome number than monocots. Autosomes and sex chromosomes are there. So the eukaryotic chromosomes are classified into two types are there, that is the autosomes and allosomes. The autosomes are not involved in the sex determination. The chromosomes which are involved in the sex determination known as allosomes or sex chromosomes. So in the diploid cells have 22 pairs of the autosomes are present in the human beings. So the sex chromosomes are usually two in number. They are the here X chromosome and Y chromosome. So there are there here the uh, here in human beings here two X chromosomes typically develop in the females and one X chromosome and one Y chromosome develop the male one. All the species do not follow the XY chromosome system in human beings. Some the differ some birds is they have the ZW and ZZ also there. Morphology of the chromosome here. The morphology of the chromosome here. The chromosome morphology changes with the stages of cell division. Mitotic metaphases chromosomes are the most suitable for the studies on the chromosome. So chromosomes, the chromatids at mitotic metaphase chromosomes are clearly visible and consist of two symmetrical structures called chromatids that they are joined at the centromere. Both chromatids are attached to the each other only by the centromere and became the separated at the beginning of the anaphase when the sister chromatids of chromosomes migrate the opposite pole. So during the anaphase, the two chromatids separated from each other and move to opposite poles. Therefore, the single chromatids represent the chromosome during the telophase. So the DNA, each chromosome is replicated during the interphase. The two chromatids produce to the replication of the single. There is a single chromatids are referred as the sister chromatids. Each chromosome consists of two types of the chromosomes. There is the one will come from the male parent and other will come the female parent. These chromatids known as the homologous chromosomes and termed as the non-sister chromatids. The primary construction and centromere. So a narrow construction that is the divide the two chromosome to two arms called as a primary construction. The centromere kinetochore is the region of primary construction where the two sister chromatids of chromosome are held together during the mitotic metaphase. During the cell division, microtubules of the cytoplasm form the spindle fibers. The microtubules are broad in the cell division, mm, the middle of the cell and concentrated at the poles. So this looks like a spindle, hence the spindle apertures, the spindle fibers attached to the centromere and help to chromosomal movement during the anaphase of the cell division. So the chromosomes contain only one centromere are known as the monocentric chromosomes. The chromosomes are two centromeres are there that we call as the dicentric, whereas the eccentric chromosomes are without centromere. So based on the location of the centromere, the chromosomes are here. Different types are there. What is see here? This one is the types of the chromosomes here. The metacentric chromosomes, V-shaped chromosomes, in which is the centromere lays in the middle of the chromosome, so that there are two arms are almost equal in size. Acrocentric centromere is located at the subterminal region of the chromosome. The acrocentric chromosomes appear as a J-type structure, J-shaped structure. Submetacentric, here the centromere is located slightly away from the center. Such chromosomes appear as the L-shaped structure. And telocentric structure, the centromere is present in the terminal position of the chromosome. They consist only one arm, hence it looks like a high-shaped structure. So this is the ultra structure of the chromosome here. Um, the centromere is made up of the heterochromatin, forming the disc-shaped structure. The cross section of the kinetochore shows the trilaminar structure. It is consist of the outer dense proteinaceous layer, a middle low dense layer, and a dense inner layer tightly bound on the centromere. The function of the kinetochore is to provide the attachment of the microtubules of the chromosomes. So spindle fibers help to in the chromosomal movement during the cell division. It also serves as a nucleation center for the polymerization of the tubulin. The protein used to formation of the microtubules. So the telomere. The repeated sequences at the end of the eukaryotic chromosomes are called as the telomeres that form a couple at the each end of the chromosome. They play the critical role in the chromosome maintenance. They protect chromosomes from the nucleus and prevent the end of the chromosome from fusing with another one. If the chromosomes break down and broken at the end of jelly, it each other due to the lack of the telomere. So secondary construction, the other than primary construction, the chromosome may show the construction and other both arms of the chromosome. So here the during the interphase, this area is associated with the nucleolus and is found to participate in the functional nucleus. It is therefore known as the nuclear organizer region or the secondary construction. The terminal part of the chromosome behind the secondary construction is called as a satellite. The satellite may appear as a round or elongated lobe. It has constant the shape size of the particular chromosome. The chromosome with the satellite is known as a sat chromosome. So the nuclear organization of each sat chromosomes contain the several hundred copies of the genes coding for the ribosomal RNA. So chromomere chromosomes during the first prophase they may show so small bread-like structure called as the chromomere. The distribution of the chromosome is highly characteristic and constant. Homologous chromosome shows the identical pattern of the chromosome. So there is another one is the karyotype. So karyotype here, this one is the karyotype here. So karyotype, a complete set of the all the metaphase chromosomes in a cell called as the karyotype. It describes the chromosomal number, size, position, and other centromeres. So here, the length of the arms, binding pattern, and secondary construction, satellite, etc. The karyotype provide the genome divided Spanish of the an individual's chromosome. The karyotype is prepared from the mitotic cell of the different tissue type that to have arrested the metaphase or all of you. Please mute. So, so the karyotype was generally presented in the form of the diagram. The homologous pair that chromosomes arranged in the decreasing order is keep mute. Hello, keep the mute. Please keep all of you keep the mute.
మర్చిపోయినా అది ఆయన అలా వేడికి జరుగుతుంది అంతే అది అది అంత ఇట్లా పూర్ పూర్లాగా వస్తుంది అది అది తీసేస్తారు అందుకే ఇంకా
The diagrammatic representation of the karyotype that is species is called as a ideogram. Generally, in ideogram, the chromosomes of organisms are bordered according to the size and position of the centromere. It shows the diagrammatic or graphic representation of the karyotype shows the number, size, and morphology of the chromosomes. So karyotypes are mainly two types are there, that is symmetrical and asymmetrical. A karyotype shows all the metacentric chromosomes is called as a symmetric karyotype, whereas a karyotype with a large difference in size of chromosomes having few metacentric chromosomes is called as a asymmetrical karyotype. The primary or advanced nature of an organism can be determined by the karyotyping. It is through the symmetrical karyotype represent the primitive state from which is the advanced asymmetric karyotype. Okay. So another one is the heterochromatin and euchromatin. So chromatin is complex of the DNA and protein. The chromosomes are formed and the basis of the staining of the chromatin with the basic dyes. Chromatin is classified into two types are there, that is the heterochromatin and euchromatin. The tightly packed form of the DNA chromosomes are called as heterochromatin. The dense of DNA is highly appears as a darkly stained region. It is found on the periphery of the centromere and telomere and nuclear organizer region. Since the DNA is compactly packed, they are genetically uh, inactive and are not transcript. So means here there are the facultative heterochromatin and constitutive heterochromatin are there here. Facultative heterochromatin means the euchromatin has undergoes heterochromatin is called as the facultative heterochromatin. The DNA of the facultative heterochromatin is specialized inactivated at the certain stages of cell cycle, but it has the potential to decondense and become a genetically active. Constitutive heterochromatin, it is consist of DNA that is the compactly inactive and remains in condensed state throughout the cell cycle. Another one is the euchromatin. So the loosely packed form of the DNA is chromatin is called as the euchromatin. The density of the DNA is low, hence it is stains lightly. It is found in the inner body of the nucleus and consists of genetically active DNA. It is the involved replication and transcription. So chemical composition of the chromosome. The major chemical components of the chromatin chromosomes are DNA or RNA. There are the here some proteins also there. So what is the DNA here? DNA constitute about the 35 percentage of the chromatin is one of the important compounds. It helps to transfer of the genetic characters from one generation to other. The DNA measured the terms of picograms. In the chromosomes, the DNA occur in two types of the unique DNA and repetitive DNA. Unique DNA, the DNA segment is consist of the single copy of the base sequences in a cell. It will not have any repeated sequences. The unique DNA contains genetic information in the form of the structural genes that control the phenotype traits. Repetitive DNA, the DNA base sequence are present in the multiple copies. So it may be either highly repetitive or moderately repetitive. Highly repetitive DNA sequence are present in more than 10 angstroms or 100 around copies. That is the highly repetitive DNA sequence are located in the heterochromatin region of the chromosome. Moderately repetitive DNA, which is the around occurs to 10 to the power of 3 to 10 to the power of copies per genome. Another one is the proteins. So the proteins here consist around 60% of the chromatin. The proteins in the chromosomes are two types are there, that is the ketone or basic proteins and non-histones. Histones as the major structural protein found in the eukaryotic types. These are all the small basic proteins with a high content of the basic amino acids. For example, arginine and lysine have the not positive changes in the facility binding the negatively charged DNA. So that is some estrogens, example H1, H2A, H2B, H3, and H4. These are the estrogen proteins. Are there. Non-estrogens, histones means non-histone protein are usually acidic and likely to bind to positively charged histones in the chromatids. There are the large number of non-histone proteins constituting the enzymes and proteins involved in the replication and transcription and regulation of gene expression. Also structure of the chromosome. When we are seeing the electron microscope and DNA proteins on base universal structure, we can explain. So here folded fibre model. Here in this model, we are proposed the Dupran Duprov in 1965. According to this model, each chromatid will have a long thread-like DNA. This model explains the DNA fragments undergoes coiling and forms around 200 to 250 angstroms of thickness chromatin fibers. So the coil of the chromatin fibers are stabilized by proteins according to this model. The extensive folding of the chromatids occur in the following ways are there. So here 20 angstrom DNA double helix is packed in the protein to form the fibril. This fibril further coiled to form a fibril of the 100 angstroms. This is fibril is called a type A fibre. The packing ratio of the DNA in the type of A fibril is at about the 6 into 1. So nucleosome solenoid model. This model proposed by the Karnberg and Thomas. So it is widely acceptable nucleosome basic model. The combining uh, the DNA and 8 Eastern proteins core is there. Here 8 Eastern proteins are there. So here 6 nucleus are coiled together and the stack are one of each other. This structure is often compared to thread wrapped around the pool. So the nucleosomes are folded through the series of the successively higher order structure to form the chromosome. So, yeah, according to this model, the nucleosome is disc-shaped structure as absent, 11 nanoarmstrongs in diameter and consists of a core particle of histone proteins and small spacer or linear DNA. So here core particles are there and spacer DNA is also present. So function of the chromosomes. The major function of the chromosomes is carry the genetic material or genetic information from one generation to next generation, and the structure of the chromosomes to ensure that the DNA is tightly wrapped around the proteins. It protects the DNA from begins damage during the cell division. So special types of the chromosomes also there. In the different form norms of morphology and function, they termed as a special types of the chromosome. That is the gained chromosomes, and another one is the break chromosomes. Gained chromosomes. A- So here gained chromosomes are in the some cells at certain stages in their life cycle contain the large nuclei with a grained or large sized chromosomes are there. There are two types are there. That is the polytene chromosomes and Lambrecht chromosomes. So polytene chromosomes were first observed in the Balbini in the nucleus of the salivary glands of the Chironomus. The larva, that is the insect larva, is there where he could recognize them the chromosomes. So these chromosomes were also called as the salivary gland chromosomes, as they were found in the salivary glands of insects. So the name of polytene chromosomes is suggested by the Kolar. So here the polytene is achieved by the replication of the chromosome DNA several times without nuclear division. So and resulting the daughter chromatids do not separate but remain alternate side by side. When a polytene chromosome is treated with a stain, it is exhibits a distinct pattern of the transverse binding. It consists of dark colored bands alternate with the light color interbrands. Okay. The chromatin of the polytene chromosomes gives an outer and a series of the loops. So a puff is band in which is the DNA unfolding into open loops are consequences intense genes in transcription. Some regions is displayed the large puff than other. These large puffy regions are called as the Balbini rings. So another one is the Lambrecht chromosomes. So the Lamprecht chromosomes, these are the special type of the chromosome found in the oocyst nuclei of these animals, which are have a large yolk egg, such as fishes, amphibians, and reptiles and birds also. So they are also found in the plants and invertebrates also. Okay. So the Lamprecht chromosomes is consist of many main axis and lateral loops also there. There are four chromatids are um, of the meiotic bivalent are held together by chiasmata in the even the large axis. The chromonema of these chromatids give the out of fine loops at the lateral side, giving the appearance of the Lamprecht or test tube brush. So a Lamprecht here, these loops occur a pair of the one originating from each chromatin. They are the surrounded by an halo of the ribonucleoprotein. Each loop has an axis which are made up of the single DNA molecule, that is the unfolding form of chromosome for the intensive RNA synthesis. So the loops provide the large So these loops provide large surface area for the synthesis of messenger RNA. The main function of these chromosomes is to take part in the protein synthesis. So another one is the B chromosome here, B type of the chromosome. It is certain plants and animals in addition to normal chromosomes, so one or more additional chromosomes, where the observed is known as accessory chromosomes or supplementary chromosomes. So these are very small and genetically invert made up of the heterochromatin. So generally, this presence in the nucleus does not affect in the phenotype, but also there's too many to they are reduced in the fertility and vigor. So this is the chromosomes and another topic is the extranuclear genome. What is extranuclear genome here? See here. This is the extranuclear genome. The, so the nucleus in the important organelle while this carrier genetic information. These genes are generally present on the chromosomes where they are enclosed in this nucleus. These genes are referred as the nuclear genes. Sometimes certain cell organelles like mitochondria and chloroplasts contain the own genes. So also bacteria also possess the extranuclear structure called plasmid. So these genes which are the present and outside the nucleus and constitute the extranuclear genome. The genes are present on the extranuclear genes are very important for proper cellular function and replicate independently of the nuclear genes. So what are the mitochondrial DNA? The mitochondria are the cellular organelles within the eukaryotic cell that produce the ATP energy. Therefore, the mitochondria also called as the powerhouse of the cell. So each cell contains the hundred to thousands of the mitochondria, which are the raw-shaped structure and the cytoplasm are surrounding the nucleus. So all the most DNA is packed in the chromosomes with the nucleus mitochondria and also small amount of their own DNA. So, what is the mammalian mitochondrial DNA? The nucleated sequence of human mitochondria was first documented. The mitochondrial DNA is a small circular DNA molecule. It varies length around 16.5 kilodalton to 100 kilodalton sometimes. The mammalian mitochondrial genome is closed circular double-stranded DNA molecule around 16.6 kilo Dalton is there. Hmm? The mitochondrial DNA is encoded polypeptide of the all subunits of the enzyme complex of the oxidative phosphorylation. The mitochondria are not self-supporting entirely, but rarely heavily for their function and important nuclear production. The mitochondrial DNA is relatively small amount and easy to isolate DNA molecule. The strand of the mitochondrial DNA can be separated on the basis of the guanine and thymine base composition, which is a result in the different densities for each strand. One of the strand is referred as the heavy strand, and this organization of the mammalian mitochondrial genome is highly compact and displays the extensional genome of the organism. The genes that lacks introns and some proteins genes are overlapped. Some of the genes does not have termination codes. The mammalian mitochondrial DNA exhibits some derivations in their genetic code to form the nuclear DNA. For example, UGA is used as a codon for the tryptophan in mitochondrial DNA, where it is code for the termination in the nuclear DNA. In mitochondrial DNA, AGA acts as a termination code, where it is code for the arginine in the nuclear DNA. The mammalian mitochondrial is exhibits the inherited exclusive to the female germ life. Therefore, it is known as the maternal inheritance or cytoplasmic inheritance. The inheritance of some characters is governed by the mitochondrial DNA. The example of mitochondrial inheritance include the cytoplasmic male sterility in plants and um, Pokinensis in the neurospora and petitized mutations in yeast. So another one is the chloroplast DNA is also. So these chloroplast DNA and mitochondrial DNA. These are the best examples for the uh, extranuclear genome. So the chloroplasts are the important cell organelles present in the plants and cyto for the photosynthesis. The higher plants exhibits lens-shaped chloroplast, their cytoplasm. The chloroplasts are plastids with a double membrane structure, which are enclosed with the stroma, thylakoid, and grana. The stroma contains ribosomes, proteins, and several enzymes which are necessary for the vital cellular processes. Chloroplast also contain a small amount of DNA in the stroma known as the chloroplast DNA. The chloroplast DNA is comparatively large, circular, and donated as the chloroplast DNA. So these chloroplast DNA is known as a plastome. The size of the chloroplast DNA ranges between the 120 to 155 kilodalton. It is around 142 in higher plants and less size in the lower plants. So the chloroplast contain about 10 to 100 copies of the circular DNA. Okay, that is the inverted repeat genes are there. These inverted repeat series of the genes into short single copies and large single copies regions are there. So here the chloroplast DNA consists of 25 genes coding for the RNA and 27 genes coding for the proteins. The proteins are mainly involved in the chloroplast gene expressions. The genes codes for the proteins of thylakoid membrane and another 10 genes are electron transport process. The chloroplast ribosomes contain about 50 ribosomal proteins and distributed between two subunits. The ribosomes of chloroplast cause the sedimentation and coefficient of the 70s um, seabird units are there. So that is 70s that we call considered as made up of the 23s and one 15s RNAs. The presence of 70s ribosomes in chloroplast, which are they resemble the prokaryotic ribosomes. So the chloroplast DNA is not associated with the histones, therefore it exits an active fibril within the organelles. The chloroplast DNA is transcribed and translocated with the chloroplast. The replication of the chloroplast DNA is semi-conservative type and also controlled by the nuclear genome, while the plastid of RNA is produced by chloroplast DNA itself. The structure and function of the chloroplast are controlled by the chloroplast DNA and nuclear DNA. So the chloroplast DNA mainly coded for the enzymes is related to the photosynthesis and protein synthesis. It also coded for the DNA polymerase and RNA polymerase enzymes. Some genes of chloroplast DNA is also coded for ribosco and most abundant proteins and F. So chloroplast DNA is mainly inherited in the majority of the plants, but not all these. Some the example of the chloroplast instances are there. So this is the some examples of the mirabilis jalafa of the leaf variation due to the chloroplast mutations in other plants. So what is the summary of the here? Uh, the organelles like mitochondria and chloroplast contain their own genome. Since these genomes are present outside the nucleus, so referred as extranuclear genome. The DNA located in the mitochondrial matrix, which is called as the mitochondrial DNA, which all denote the mitochondrial DNA. This is the first documented mitochondrial human mitochondrial DNA. So the chloroplast DNA is comparatively large, circular, denoted, which is as a cpDNA means chloroplast DNA. It has to 10 to 24 kilodalton and long sequences present two distinct copies per gene and inverted repeating.